Dr. Claudia Boot of ARC, expertise in both NMR and mass spectrometry. So we are looking forward to her presentation. And I just muted one of the participants. FYI, that was me. I apologize. So I'll turn it over to you, Claudia. Thanks for having presenting for us.
Thanks, Corey, and everybody for coming to this talk this afternoon. My primary goal is to help you understand metabolomics offerings in the ARC and really focusing today mostly on NMR metabolomics. It's a new offering for us, and I'm excited to tell you more about it. I'm going to couch it in the context of this story about exploring Arctic soil carbon molecular structure and how it's responding to global change factors. But before I do that, I'm going to start by reminding everybody about how carbon gets into soil in the first place. So plants are the primary way that that happens. They pull CO2 out of the air and put it below ground in the form of different types of organic molecules. Those materials are often degraded and released back into the air through root and microbial processes. And usually this is a balance flux. So generally the same amount of carbon that's going into the soil eventually comes back out of the soil. And this is sort of a zoom in on that microbiome influence on soil carbon cycling. We know that the processes that result in carbon efflux out of soil are temperature dependent. And we also know that our planet is warming. And there's kind of two alternative scenarios with global warming where you can imagine a positive feedback where global warming causes some change and then that speeds up warming and then that cycle goes around and around. Or a negative feedback where global warming causes some change that actually slows down the process of warming and creates a negative feedback. So that's like in a general sense, a way to think about these feedback loops. In the case of soil carbon cycling, there's kind of two ways to think about these two alternative possibilities. One is that rising temperatures causes soil temperature to increase and the amount of CO2 that's coming out to increase, feeding back positively to net higher temperatures on the planet. But another possibility is that rising temperature can cause greater plant growth along with fertilization of that additional CO2 from the atmosphere. And plants may draw down more of that CO2 than that's coming out of the soils, which could result in a negative feedback and overall lower net temperature changes. And we don't really know, depends on the soil system and a whole lot of other variables, how these feedbacks are working in different biomes. The biome I'm going to apply this to is in the Arctic. And the Arctic is cold, so generally decomposition is really slow because temperatures are below zero for so much of the year. And we also know that there's a massive pool of carbon in the Arctic. This is a scale figure for different sizes of the carbon pools on the planet, and almost half of soil carbon is stored in permafrost soils in the Arctic. So this feedback is really important to understand how temperature is going to influence carbon cycling in the Arctic. The other thing we know about the Arctic is that it's like the rest of the planet getting warmer, but it's getting warmer faster. And it's warming at least the double global average since the year 2000. So this is a really critical feedback to understand in this ecosystem. We also know that with these warmer temperatures, we're altering seasonality in the Arctic. So spring is coming sooner, summer is lasting longer, and the plants are responding to that change. So there's an increase in, this is measuring peak greenness in Arctic ecosystems. And this is really caused by shrub expansion into different areas in the Arctic. So plant community changes are happening with this altered seasonality. So that's the setup, the framework that I want you to think about as we're going through this data, this research question. And it's really, I'm posing it as, is it vegetation type in the Arctic? So I'll introduce those in a minute, but shrubs, tussock tundra, and kind of swampy wet sedge tundra. Does it matter that those plants are expanding, or is it this altered seasonality that's influencing the soil carbon molecular structure in the Arctic? So the setting for this study is here at the Toolik Field Station, located just north of the Brooks Range, above the Arctic Circle in Alaska. And if we zoom in to what the field station looks like at Toolik, we have the lake here. And then these colors are going to carry throughout the talk, where in green we have tussock tundra. This is Eriophorum vaginatum. It's like probably the most charismatic plant that I've ever encountered. It's very adorable. And in red we have the expanding shrub tundra. This is Betula nana. And in blue is a wet sedge tundra. So this is like a swampy, grassy lakeside vegetation. And this is showing, I'll talk about methods in a moment, but this gas-powered ice auger that we use to collect soils when we're there in the winter when temps are below zero. So the soil collection for these samples for the first part of this story took place in 2010. And here I'm showing temperature on the y-axis and date on the x-axis. So you can see surface temperature in blue and subsurface at 10 centimeters in red. As the soils begin to warm in May, then we have our growing season here, and then they return back below zero in late September. And our sampling for this study took place, I'm going to just call them rather than months, seasons. So late winter, thaw, late summer, and early winter. So kind of over these temperature transitions at the start and end of the growing seasons. And these colors, so it's going to be complicated. You can tell there's colors for vegetation and colors for season, and these colors will carry throughout the talk. This shows you a little bit of what the tussock tundra looks like. It's kind of undecomposed plant roots is what we call soil for this type of vegetation. These are all collected, and we measured biogeochemical variables, pH, water content, carbon and nitrogen pools by centrifuging the soils, subsections of the soils, centrifuging the pore water out of them. So this is the water that was actually trapped in the soil core that we're pulling out of them through centrifugation. And then we also measured the metabolome of that pore water. In this study, I'm going to talk about briefly some LCMS data from 2010, and then we'll spend most of the talk focused on the NMR metabolomics data that we have from 2011. So the biogeochemistry first, so here's everything together. Now we've got our 3 vegetation and our 4 seasons, and you can see some dynamics in pH and water content over the changes in those seasons. And those are coupled with the sizes of, this is the carbon pool and the nitrogen pool. And we see this general pattern that is pretty consistent throughout the Arctic, where there's a large amount of carbon flush during thaw, carbon and nitrogen, that really disappears by the end of summer. It's all been decomposed. And then it starts to rebound after the soils have frozen again. So this gives us a snapshot of our biogeochemistry. And now we're going to dig into the metabolome. What's actually the molecular structure of those pools in this system? And this is just a generic workflow for any untargeted metabolomics project, where you would begin with these biological samples. In our case, it's soil pore water, collect some kind of spectral data. In this case, it's LCMS. and then process that data to remove noise, align peaks, and deconvolute the spectra to generate, in the end, a feature matrix. So you have your M over Z values if it's mass spectrometry for each sample and their intensities in a data matrix that then you put through a data analysis workflow to generate univariate and multivariate results, and combine that with attempts at metabolite and annotation and some kind of biological interpretation. Can you do a network analysis or a pathway analysis? In practice, these first 4 steps are very straightforward. Things get more difficult with metabolite annotation and biological interpretation, and we'll talk a little bit about that closer to the end of the seminar. So, results in a nutshell, that by LCMS, we could see within each season, you could distinguish those 3 vegetation types, the molecular properties of those 3 vegetation types from one another. It's most clear in the late winter thaw and early winter, and over the course of the late summer, that carbon starts to become more homogeneous and less distinct among those different vegetation types. But the real story is that no matter where you get the carbon, season is the main predictor of Arctic soil carbon metabolite profiles. Here, this is all of the vegetation types together. We start in the late winter thaw. There's a very small change in composition, and then pretty dramatic over the course of the summer, and even more dramatic after the soils freeze in the early winter. And this we can visualize using, this is just a heat map showing each individual sample on the bottom. And these are all M over Z values and retention time. So analogs to molecular weight and some indicator of how polar the metabolite is. This is early days for us for me and my metabolomics journey. And so we didn't attempt annotation with this information. We just suffice it to say that we can tell the differences in these profiles over the course of these seasons, and that it's really season that's more important than the vegetation. So that's sort of your intro into this study that was done with LCMS. And now we're going to turn to using NMR to look at these molecular profiles. And there's a few reasons for doing that, but I like to think of NMR, I say NMR is the truth. Like if there's a proton on your molecule and you put that sample into a magnet, you're going to see that proton. It doesn't depend on the molecular structure, where with mass spectrometry, if something ionizes well, you get a whole lot of signal. And if it doesn't ionize well, you can't see it at all. And so you don't have, you have an idea, you approximate the truth, but you don't really know the truth when you're using LCMS. The drawback is that NMR is far less sensitive than mass spectrometry. So you're going to get gross structural kind of bulk chemical changes, but you're not going to get the fine level of detail and an annotation available to you with a mass spectrometry data set. So you're maximizing with NMR certainty as well as low cost and high throughput. I think samples are around $5 a piece. You can run a spectrum in 10 minutes with the way we acquired them in this experiment. And you're trading that off for the sensitivity and coverage that you get with LCMS. So we took NMR as an analytical platform to look just over in 2011, this thaw period. So this was an intensive sampling. This is at when the soils are below here, they're frozen below 0. And I have 6 dates across this thaw sampling in 2011. Today, I'm just going to talk about the late winter and early summer, the bookends of this date, of this sample field campaign. And we have, just like I did in the last example where we used LCMS, we have soils from late winter, early summer, but we also have not just the soil pore water, but a microbial dissolved organic matter pool. And the way we pulled this out is we took those cores, we split them into pieces. Half of them we fumigated with chloroform gas for 4 days before we extracted the pore water. So we broke open all of, or many of the microbial cell membranes, and we can actually see the intracellular carbon pool and how microbes are metabolizing carbon over the course of this thaw period. And we wanted to know, with this sample set, can NMR give us a similar level of information as what we found from mass spectrometry, both in terms of that vegetation question and that season before and after thaw in the case of this sample set. So these samples were all acquired on our Brooker Neo instrument that's equipped with a Prodigy cold probe that really improves sensitivity of NMR. They're all normalized to an internal standard. We acquired them with quantitative acquisition parameters, processed the spectra in MNOVA, and analyzed it in Metabolomics. So this is what the spectra look like. This is showing just late winter, the late winter date for both the soil pool and the microbial pool together. Now, if you aren't used to looking at NMR spectra, this is a helpful diagram to kind of orient you to what you're getting, what information you're getting from this spectra. So the position, the chemical shift of your signal on this axis corresponds to some functionality. And it's very consistent across any samples where all of your alkyl carbon is going to show up upfield in this region, and all of your, say, aromatic carbon is going to show up down in this around 8 region. So already you know more from this than you can gather from just having an M over Z with your mass spectrometry data. It's not better, it's just different. So this is really useful, though, that you can get structural information. And when we process these data, this is using Metabolomics, where we feed in our data matrix and then do some univariate statistics. This is just an analysis of variance that's telling us the features are just numbered down here. After filtering, we kept 5,000 features, and about half of them were considered significant for the comparison between the microbial pool and the soil pool. So the darker red and the larger the spot, the more different they were in those two pools. And if we, choose this most different spot, this is what the output looks like, where the intensities, this is on a log scale, of the fumigated or the microbial pool are all much greater than the soil pool. This isn't terribly surprising. We would expect the microbial pool to be different than the soil pool, but it's a nice proof of concept that, yes, okay, this works. We can just see differences in these two pools. And when we look at the same data using principal components analysis, this is now the microbial pool in the red, blue, and magenta, and the soil pool. They clearly separate from one another. And when we dig into what the chemical shifts are, where we see these differences, it's pretty obvious when you look at this spectra. You can see there's a whole lot of resonances in this microbial pool that show up in this 1 to 2 region, and then also out at 2.5. And these correspond to things like membrane lipids, right? You broke open microbes and you get out a lot of their cellular contents, but also their membrane contents are showing big differences. So I kind of walk through this slowly as an example of how you work through the data and then how you use what you pull out of the statistical analysis to go back to the spectra and say, well, what are these things? This is a pretty obvious case in the case of microbes versus soils. So now we'll get into our question about seasons and vegetation in the Arctic. Can we pull out those types of differences using our NMR data? So now what we're looking at is the same type of plot with the ANOVA for a comparison of just the soil pool. So now I'm not going to compare soil and microbes anymore. We know those are different. But we want to see within the soil pool between late winter and early summer, can we see differences? And the answer is yes. About a third of the features of this filtered data set showed up as significant in this ANOVA. And if we look at one of the significant features, we can see, okay, this isn't terribly surprising. We see greater intensities late in winter, and as summer comes through, this intensities are coming down. But what is interesting, um, yeah, so we're still kind of saying the same story that season is the real predictor of composition of the soil organic matter, and late winter generally has more material, more of this type of material than in early in the summer. And the ordination isn't as clean, but we still see some pulling apart where our um, late winter samples are separating some from our early winter samples. And we can do a biplot of these to identify resonances that are contributing to this separation in ordination space, and then also compare that with our ANOVA data to say, all right, do we have overlap in our statistical tools? And they're indicating that, yeah, in this region, 1.15 and 4.2, we've got significant differences happening in these two pools. If we look now, instead of at the soil pool, but at the microbial pool, again, we see fewer differences overall between the two seasons, but still a large number of significant features that are changing in response to season. And again, in the aliphatic region, this looks the same as what we have seen in the other plots, where we see a reduction in this molecule after the transition to summer. But what we start to see, and that comes out in our other significant, most significant variables, this is the chemical shift here, we start to see this pretty nuanced difference where in the early summer, some of this material is still elevated in our shrub tundra, and you can see a large change across vegetation type early in the summer. Whereas late in winter, they were basically the same across all pools. And that also bears out for some of the material we see in the aromatic region, where we see this stratification, and generally it's shrubs that have more material early in the summer than what we're seeing late in the winter, and a consistent concentration across all of the different types of vegetation. So, how do we interpret this? How do we kind of link back to our spectra and dig into what we might learn based on the chemical shifts that are significant in our uni and multivariate analysis? And what we can see is here I'm showing you a subset, two of each type of vegetation of the microbial pool from late winter and early in the summer. And when we looked before at just like the microbial pool versus the soil pool, it was obvious what the differences are. Once you stack all of these spectra together, your eyes do not tell you the story. Like you can't see it anymore. You have to use the statistical analysis and then go back and look at your spectra and say, aha, here this is. This is a really clear signal. It looks like a triplet that's been changed to a doublet and shifted a little bit downfield. I didn't dig into what molecule I think this might be, but the way you would go about that is to look at the chemical shift of this material. So we're in that alkane, aliphatic carbon region. Think about the multiplicity and connectivity of that hydrogen. And then you probably use some two-dimensional NMR and other regions of the spectra to figure out what other carbons are the hydrogens on this carbon connected to. You can also use your metabolomics analysis to infer some of that. So this is awful looking. You can't read anything. You just see big red splotches. But what you're seeing here is the NMR spectrum, just chemical shifts from beginning to end. And this, where it's bright red in the middle, is where the shift matches itself, right? So that's a high signal. But what we're seeing here is like in this region, all of these resonances shift in the same way with their intensity in your data set. And so you can say, okay, I pulled out, maybe this is what my statistical analysis pulled out, but the regions that are changing, like here, are changing in the same way altogether. So those protons are related to one another and how they're responding to the environmental question that you're asking or whatever your study system is. So you can use two-dimensional spectra. You can use this correlation analysis to dig into what these things might be. And I also think what I would suggest people do, which is often not done, is to use mass spec and NMR together. I think NMR makes for a really wonderful way to pre-screen your data and look for these kind of like very clear, very interpretable structural changes based on your research question. You know something about the molecular functionality and the structure, you can use that to guide maybe the chromatography you'd like to use for mass spectrometry and what type of functional representation you're looking for in things that are significant. So if you could correlate your results from both analytical tools together, you'd have a lot more certainty that you know what's going on in your system. And I would say the other kind of outcome that's not relevant to the experiment, but to the technique is that I found the NMR to be surprisingly sensitive to these shifts in microbial dissolved organic matter composition in a way that I wasn't really expecting. I thought, sure, we can tell microbes from soils, and maybe we can tell even a low amount from a high amount, but the within vegetation changes that we're picking up with the NMR, I think is impressive and a good reason to explore this as a useful technique, even in systems where maybe there aren't clear-cut differences. So to kind of wrap up the research part of this story, I posed this question at the beginning is it vegetation type or season that's a better descriptor of the soil carbon molecular structure? And if you weren't following any of that, that's fine. The answer is season. Okay, so this is what's more important. Eventually, maybe the shrub expansion will become more important, as we did see there are stratification in different compounds by vegetation, but the real driver is season. And unfortunately, I think that puts us in the category of this first scenario where this longer growing season and this gigantic pool of carbon that's available in the Arctic is likely to result in an increased soil temperature and an increased efflux of CO2 from soils, resulting in that positive feedback for climate change. Um, so that's the bad news. I don't know, is there good news? Let's see. Okay, well, thank you to everybody. On the research side of this, I had a lot of collaborators on this project. I also want to especially call out Michelle Melhot. She's the NMR spectroscopist here in the ARC and was instrumental in getting acquisition parameters and sample preparation set up correctly to do this type of research. And the NSF provided the funding for the field campaigns. And so you might be now asking yourself, this looks really cool. How do I do NMR metabolomics in the ARC? And as all wonderful things in the ARC, you begin with requesting training in iLab. Michelle will train you to use the NMRs, and then you'd probably have a conversation with us about identifying an appropriate internal standard for your study system that you can use to reference your spectra and normalize the results to. You want to avoid resonances that overlap with parts of your spectra that you're interested in and need to consider solubility of that material depending on your study system. You also need to think about sample preparation in terms of how much material you have. You need a few milligrams in order for this to work at a minimum. You can't have things be too salty or have a lot of metals in them. So people that work on soils, these are definitely things you would need to consider in order to do this type of research. And often you want to buffer your sample to a consistent pH if you have, like we did in this study, very different pHs of that pore water to begin with. Then you'll acquire your NMR spectra, and Michelle can help with the experimental setup for those samples. And then you dig into the data processing. We have the tools we need here at CSU. Mestranova, we have a site license for through the chemistry department that's available to all users of the ARC. And using that processing software, you can do your phasing and baseline corrections, referencing and normalizing your spectra, and basically arrange everything to export them in that data matrix that then you can input to a program like Metabolomics, which is a web-based free program, or R if you're savvy. Everything in Metabolomics is done in R, and they publish the code alongside of the processing. So if you want to take that and use it in your own processing workflow, you can do that. So there you're basically, labeling your experimental treatments, you're filtering that matrix for misring data and removing redundant data that's not contributing to detecting differences, transform and scale the data through that program, and then apply the uni and multivariate statistical analyses. So some of the figures that I showed you today are generated in Metaboloanalyst. You can also use Metaboloanalyst for your mass spectrometry metabolomics, so it's nice to have one processing software that can handle different types of spectral data for similar types of output. And then you're left with identifying, what are your responsive resonances and what's the structural information that you can draw from those samples. And that may be something as limited as functionality, or if you have standards for things that you think will be changing in your experimental system, you can acquire NMR on those standards and compare your experimental spectra with your standard spectra and say, yes, these 4 molecules are changing in response to my experiment. So I will just wrap up here by saying we have a few options for self-service metabolomics in the ARC. We run a training course a couple times a year for mass spec metabolomics. That takes place over 2 weeks, just in 4 different sessions. We'll be offering that again soon. And for now, with our NMR metabolomics, we are happy to be a consultant on, designing that experiment and helping you set up acquisition parameters and walking you through data processing. So we're here to be resources for anybody that wants to explore that in their research. And I'll also just mention that we were awarded an NSF MRI grant for a 600 megahertz NMR that will also be equipped with that Prodigy cold probe. So we'll have really nice sensitivity and improved resolution of complicated regions of the spectra that we'll be able to use in this type of research. That should be coming online late this summer. There's a QR code to fill out an interest form if you're interested in the mass spec metabolomics training workshop. And I will just wrap up with these conclusions that we were able to show LCMS and NMR metabolomics provide nice insights into the molecular structure of soil carbon. Using mass spec, we showed that seasonality is more important than vegetation for altered carbon cycling under global change, and that the NMR data supported our mass spec conclusions and was surprisingly sensitive to intercellular pools of metabolites within each vegetation type, and that the ARC is happy to support your NMR metabolomics research questions. Just send me a note or reach out to Michelle, and I'm happy to take any questions. Let's see here.
I see a hand up from
Amelia? Yeah.
I'm Amelia. I'm in the Bork lab. I had a question about your internal standards. So for this experiment specifically, how did you go about choosing that versus other internal standards? And what was that internal standard?
So I used a deuterated DSS internal standard. These are water essentially, so it's a very polar internal standard that has a chemical shift of 0. And so it almost never is going to overlap with what is showing up in your analytical samples. It's nice because you can prepare that as a stock solution in water and just mix it by volume for a known concentration. A lot of the other studies that take place in organic solvents, chloroform is a really popular one, and you can't, it evaporates too fast to have accurate transfer ability to know the actual concentration of that internal standard. So you have to work with that. Internal standard in organic solvent is better in a solid form, weighing it out for each of your samples. But for soil, water extracts, DSS is a really great internal standard to use for that system.
Thank you.
Yep.
See a hand from Santosh.
Bring you guys up here. Yeah, go ahead.
Very interesting. I have two questions. As you showed that microbial and plant-specific metabolites. So in your analysis, did you got any metabolites just specific to plant or microbes? And my second question is that you got conclusion as a variation is because of seasons, but maybe there are seasonal variation of plants as well as microbes. How can you connect that? This variation is because of seasonal changes in microbes or plants.
So your first question was, were there any metabolites that were unique to the microbial pool? Is that correct?
Yes.
Yes. So definitely we saw when we compared just the microbial pool and the soil pool, definitely a lot of membrane lipids were showing up in our NMR analysis that we weren't seeing in the soil pool. That was the primary difference that we saw, but I think if, of those 2,500 significant features, we only looked at the handful that were most different, so we didn't dig down into what else was there. That might be interesting to see what we could pull out of the microbial pool that we didn't see in the soil pool. And then I think your second question was, my conclusion was that season was a bigger driver than plant vegetation type in terms of the overall composition of the organic matter. And I think the point you're making was that there's also a seasonal aspect to plant inputs, which is 100% true when they're growing at the beginning of the growing season compared to when they're senescing at the end. They're going to have really different inputs over the course of that time. And I think when we look, so I showed ordinations. I can't say for mass spectrometry or for NMR because we only had the two seasonal points, one when the plants weren't growing and one when they were. But with the mass spec data set, I don't think I've looked at it as how did the wet sedge compare across the 4 dates with each other. They do, yeah, so I would suspect that you're correct, that there is a seasonal aspect to the plant community, and I would just have to input it in that way to the way I set up the analysis in order to do the analysis to be able to say, but I suspect that you're correct and that there is a seasonal component to the plant inputs as well. Did that answer your question?
Yes, correct. Thank you. And I have one more small question. So
Sure.
whatever key IDs we get at the end of analysis using Metabolomics, do we need to again re-verify using standard compounds, that MSMS fragmentation?
Say your question again.
Suppose we get some KEGG IDs from the Metabolomics, which are significant variation among the samples. So do we need to validate these metabolites also again with standards? KEGG IDs.
Maybe.
Can you comment on that?
Yeah, so it's always a good idea. If you are going to go with that, with where you might call a level, I don't know, remember if it's 1 or 4, but the best type of annotation, level 1, I think, annotation. That means running an authentic standard on your analytical platform that you collected your data on, and you know that that is that molecule because you've demonstrated it on your system. Do you have to do that in order to call it that molecule? No. You can say, these spectra match. Here's the reason why. Here's my justification. It's better if you do, but it's not always possible. You might end up with, a few hundred significant metabolites. If you're fortunate enough to identify all those with KEGG identifiers, you want to have a very clear idea that that's actually what that molecule is, and you should have a good justification for it. But I don't think you have to run them as analytical standards on your own system.
Thank you.
Jacob?
Hey, Claudia, excellent presentation. I have a bunch of questions, but I'll just narrow it down to two for right now.
Okay.
The first one is kind of what DOC threshold do you need to get good data from this system? Because I was looking at your numbers and you had some samples that was around 600 ppm DOC. So what are your recommendations for, what levels that should be at? And then
Yeah.
the other question I have is how do you identify features in NMR? Because in mass spec, there's a peak in a chromatogram. That's how you can identify a feature, but multiple
Yeah.
peaks in an NMR spectrum could correspond to one particular molecule. So I'm kind of curious on how that process looks.
Yeah. It's good questions. So say your first question again.
Oh yeah, DOC thresholds. How much
Oh,
carbon
right.
do I need in my samples? Yeah.
Yeah, you need so that is a nice thing about Arctic soils is they are carbon rich. And so you have a lot of material to work with, and that's why when I was rooting around the freezer for a set of samples that would be good to develop this workflow with, I thought, ooh, I've been saving these for a rainy day and I know what I'm gonna do with them. So I think you could probably get away with 5 to 10 milligrams of carbon. I think we will push that with the 600 system, and you might be able to go to 2 to 4 milligrams of carbon. I think before you do any of this, obviously you'd want to run a few spectra, see what your NMR look like. I think there's also, I think we may be having a salt tolerant low volume probe that's coming with this 600 system that's really going to improve the sensitivity for low concentration samples, which is often the case in soil research. So I think definitely the short answer is just try it and see, like, oh yes, this looks good. Do you have a nice enough signal to noise where you can actually see differences? And then, if not, just use mass spec, though you might be limited there too. And then your next question was about, when you have mass spec, you have a feature, you have a retention time and an ion, and that seems like a discrete thing. Whereas you're right, with NMR, you have kind of a continuous data set almost. And the way this used to work is you used to have to choose like spectral bins, like you could choose a bin size of a half a ppm or something to bin your spectra. But that was more of a computational limitation than a data limitation. So I think AMNOVA defaults to something like, I don't know, it makes tens of thousands of data points out of a spectrum. And I don't bother binning those. I just dump it all into Metaboloanalyst. And you noticed in those ANOVA plots, there were like big regions that were significant. And usually those are all the same chemical shift. And so it kind of tracks out that you can see, 1.19, 1.195, 1.12, like you can see where those things are a single resonance. Now, as far as connecting that resonance with the other protons in that molecule that might also have to also be responding to whatever your experimental setup is. There, you want to go back to your data and do some 2D NMR. So if you say, okay, are these things on the same molecule? like they're responding in the same way. Are they actually part of the same molecule? And I would say you would do this regardless of whether or not you think they are, just to discover where the other protons on that molecule might be. And then you can say, okay, these things are on the same molecule or they're not on the same molecule. And you can usually tell that from your two-dimensional data. So COSY or TOXY spectra. Yeah. Does
Awesome,
that
thank you.
answer your question?
Yeah, that really helps with the DOC because I thought I was like, okay, I need hundreds of DOC or milligrams per carbon, so it's good to hear
No,
that
it's
not the case.
nice if you have
Yeah.
that, but I know that's not usually the situation.
Yeah, and it seems like features and NMRs are more kind of like very discrete chemical shift bins rather than a
Yep.
specific ion. So I just got to change my understanding what I'm classifying as a feature. So that helps a
Yep.
lot. Thank you.
Yep, sure. Santosh, did you have another question?
No, that's thank you.
Okay, just checking. Anybody else? Okay.
Well, thank you again, Claudia, and thanks everybody for coming.
Yeah, nice to see you all.